Okay, great. So what we're going to do is keep moving for my talk, so I'm going to introduce myself. Here I am. Can I give myself a bottle of wine? I don't know. And then we'll probably have some downtime as we set up, hopefully, the next talk, God and Skype willing, that Dr. Trier will be able to give live from Berlin. So you've already heard, I think, some fantastic talks updating you on our most current clinical results. And so what I want to do now is go a little bit into the realm of what seems like almost science fiction as we apply some incredible technologies, things that I never would have even dreamed of 5 or 10 years ago, that I think are going to impact on not only our understanding, but most importantly, potentially to come up with novel therapies. And, you know, it's such an incredible example of how much there is strength in knowledge. So here we go. I should also make a couple of disclaimers. One, I am not a neurobiologist, so I'm risking completely embarrassing myself. I would also like to acknowledge, actually, that a lot of these slides that I'm going to show you are provided by one of our pediatric cardiology fellows who happens to also have a PhD in neurosciences. He spoke actually at our conference 2 years ago, Andres Bradenchak, who couldn't be here today because he's moving back to Hawaii as a new faculty member there. So, but he sends you his very best wishes. So, again, as I mentioned before, the questions of behavioral and cognitive problems have come up from you guys. And so, again, this conference is all about you, and none of these talks, none of the sessions today, for example, would be possible if it weren't for the ongoing feedback and concerns that you guys have given us. So what I've listed here are just some of the critical questions that, for lack of a better explanation, I felt are what we need to use to move forward. So the most basic question, I guess, is, are there genes in the region of 11q that are deleted that are involved in brain development and function? That's a pretty obvious question to ask. And then a very important question is there a correlation between the presence or absence of these genes and certain behavioral or cognitive features? So I'll show you more in a minute. For example, intuitively you might think, and that appears to be the case, that for the most part, the bigger your deletion size, the more affected you're going to be. That's not too surprising. And then most importantly, and this is a really important point to highlight, that this isn't only for your kids, but perhaps a much greater sector of the general population that may have some of the same types of problems that your children have, and that is, are there any potential treatments or therapies that can be developed based on understanding the real mechanisms of how these genes function? So our aims are, one, you heard, for those of you that heard Dr. Delacqua talk yesterday, thanks to these incredibly recently developed techniques now that allow us to map deletions not just at a sort of macroscopic level, but down to the level of individual genes. And just to give you an idea, about 12 years ago, the first human genome sequence was published. That was a project that I think cost $3 billion to accomplish. But because of that now, we can get a readout combined with this array CGH technology of exactly what genes each of your children have deleted. And so that's critical. Nowadays, 12 years later, the cost of sequencing everybody's genes, at least at the wholesale level, is somewhere around $1,000. So that's an incredible accomplishment. And actually, one of the reasons why Dr. Matina wants to draw blood from your children is we're going to apply some of those very recently developed technologies to potentially sequence every part of every gene of your children. So as you can appreciate, before we talk about genes, we really have to understand the clinical phenotype, and that's why I can't emphasize enough. Oops, hopefully somebody didn't fall apart here. I can't emphasize enough the importance of these comprehensive characterizations that Sarah, Natasha, and her teams are performing. Without that, we couldn't make any sense out of this. And then, using these DNA technologies, we can begin to ask whether or not there are correlations between the presence or absence of these genes and specific cognitive or behavioral phenotypes. And as you know, there are limitations to what we can do in humans, although I'm going to tell you how we're getting around that. But obviously, we can use animal models, believe it or not. There are laboratories even here in San Diego that study behavioral and cognitive features in mice. And so we can do genetic engineering to potentially recapitulate in mice some of the same clinical problems that your children might have. And most recently, stay tuned for a future Nobel Prize in the next few years, and that's the reason why we have asked you to so graciously participate in the skin biopsies. It gives us an incredible opportunity to actually recapitulate human disease in a dish. And what I mean by that, and I'll show you a slide later on in the talk, we can actually take skin cells, that's why we do the biopsy, and through some genetic trickery, reprogram those skin cells, for example, to become neurons. And we can study then the function and the structure of those neurons in a test tube. And for example, as you heard a little earlier, if we think for some forms of autism that it has to do with the way neurons come together to form the specific connections, we can analyze that in the test tube using normal controls and looking at your children that either do or do not have some of these specific behavioral phenotypes. And that's a critical requirement that can potentially lead to drug development. So this work actually started quite a long time ago, I think maybe about the second conference we had here. And initially, we studied 13 children, and essentially what we found, not too surprisingly, was that there was a correlation between the size of the deletion and the degree of cognitive dysfunction. And so since that time, we've actually analyzed a lot more, but initially the next round of studies involved 14 children where we performed comprehensive neurocognitive assessments, and then we used this array CGH technology to look at that readout of the genes deleted. And so we actually published this paper a few years ago, and what it then showed was that there indeed was a very strong correlation between the size of the deletion and the degree of cognitive dysfunction. And one of the genes is a gene called BSX, or brain-specific homeobox protein, which hopefully, if the Skype works, you're going to hear the world's expert on that gene in the next hour. And what's very clear, and I'll show you some data to support this in a moment, is that if your child has this gene deleted, and virtually, I think without exception, there are some exceptions for the smaller size deletions, but anybody without exception who has a deletion that deletion to the end of the chromosome that spans this BSX gene, and what that means is about a 12 megabase deletion, that gene seems to be responsible for causing the more global, more significant cognitive impairments. So that kind of separates the big deletions from the children with the rest of the deletions. And then we think that there are subsets due to multiple genes that are deleted in the children, even with the smaller deletions, that contribute to varying degrees of some of these cognitive problems as well as behavioral problems. I hope that makes sense. So let me just say that in a little more systematic way. So the results are as follows. For at least that initial 14 patients that we studied, there was actually 100% correlation between the relative deletion size and the degree of cognitive impairment. And now having the ability to look at the human genome, when you look at the genes in there, there are multiple genes, not too surprisingly, in this region that are deleted, that are involved in brain development. And there's actually, again, this very small region that seems to be a very clear cutoff that's where this BSX gene is located, that determines whether or not your child is going to have more significant delays. And then looking at the children with the smaller size deletions, there are variations between those children as well. So that's why I sort of qualitatively divided to small, medium, and large size deletions. And we think that there's even a difference between the small deletion and the medium sized deletions. And in that region that distinguishes the medium from the small size deletions, there's a very interesting gene that I'm going to spend most of the rest of my talk discussing, called Neurogranin. But again, these are based on small numbers, and that's why it's so important to study more of your children. And so in addition to that, for those of you that heard Dr. Delacroix's talk, all the results that I'm telling you about are based on your children that have what we call a terminal deletion that goes from a certain point to the end of the chromosome. And now recently there are actually huge databases available actually to the public, as well as some proprietary databases of this array CGH analysis. And we've actually found some patients that have cognitive delays, behavioral problems, that rather than having a deletion that goes to the end of the chromosome, they have a very small, what we call an interstitial deletion. So it's within the region, but it doesn't go out to the end. And that turns out to be extremely informative because those interstitial deletions only have a very small number of genes. And because of that, that's actually how we found the heart gene that I'll talk to you about tomorrow. And so far, we have found several patients with interstitial deletions that have cognitive delays that have either the BSX gene deleted, the Neurogranin gene deleted, and actually several more that I'll mention at the end of the talk that we're very interested in. Because when you look at what these genes do, they seem to have common functions in brain development that might give us insights into the mechanism. So this is just sort of a graphic way to show you the relationship between deletion size and full-scale IQ. And really all you need to look at is, for this slide, just look at the difference between the medium and the large deletions. So the children that basically have this BSX gene deleted and that the deletion goes out to the end of the chromosome are ones that have by far the most significant impairment, at least by the way we measure IQ. So those children generally have an IQ in the 40s. I don't think we've ever seen one that has one greater than 50. And then if you go to slightly smaller deletions, less than 12 megabases, basically all of those children, most of those children have deletions, full-scale IQs that are significantly higher, anywhere from 60s, 70s, and 80s, meaning that we're talking about low normal IQs potentially in some cases. And if we force the issue a little bit, we think there are some subtle differences, which I'll show you in a moment, that distinguish those with the small versus the medium-sized deletions when looking at full-scale IQ. So again, just to reiterate, I guess I keep repeating myself, but for anyone whose deletion size is more than about 12 megabases, their IQ scores are less than 50. And that, I think, is without exception. Having said that, all patients that were relatively only mildly impaired then have a deletion size that's 12 or less than 12 megabases. And that's how we found this BSX gene. So you'll hear about that in the next few minutes. So then the question is, what about the rest of the population? So about a third of the children have the large deletion that spans the BSX gene. And then the question is, can we distinguish differences between those with, let's say, a small and medium-sized deletion? So interestingly, when you look at that original group of the 14 patients, the 3 patients that had the highest full-scale IQ had the smallest deletions. And that's not probably a coincidence. So the same 3 patients had the highest index of something called freedom from distraction or working memory. I'll talk about that in a moment. And even patients with the smallest lesions, though, as far as we can tell, either have mild or low normal, like mild impairment or maybe even low normal. So we don't see anybody, I think, that scores an average of 100. Having said that, though, keep in mind there are exceptions. So, for example, if you have a baby that's born with that has a small deletion, and let's say they have a brain hemorrhage because they were born prematurely, obviously there are many other factors that can cause cognitive delay, so there's not a 100% correlation. And there certainly can be other genetic factors besides what's on 11q that might cause potentially more significant delay than what one might predict for a smaller deletion. So this is an example, if you look at the difference between the small and the medium-sized deletions of a specific aspect of learning called digit span, where you're given a list of numbers and then you're asked to recite those back. So it's a type of memory function. And actually, again, our numbers were small, but we actually see a pretty dramatic difference between those with the small versus the medium-sized deletion. So the scale on the left is just sort of an arbitrary scoring system, but the higher the number, the better they do. And that's what then implicated this gene that I'm going to spend most of the rest of my talk telling you about, called Neurogranin. And so Neurogranin is a gene that's deleted only in those patients that have a medium or large-sized deletion. And there's actually a lot known about neurogranin based in animal models. It's critical for something called long-term potentiation, which is a form of learning. And I won't go into all the biology. As I said, I'm not a neurobiologist, but neurogranin actually interacts with a protein in the brain, in the neurons, that stores something called, well, you've heard of it, calcium, and calcium turns out to be a critical mediator of learning. Calcium is the basis of these electrical currents that actually constitute learning and memory. And so basically in mice, the more neurogranin there is in the brain, the stronger that signal is. So we think also, interestingly, that, and I'm going to show you an example of this in a moment, that for those of you that may have heard of something called environmental enrichment, a lot of your children you're probably told, do everything you can to stimulate your child. And it turns out that neurogranin seems to be actually the molecular mediator of this process of environmental enrichment. And that's fascinating because environmental enrichment is actually recommended for people with a lot of different kinds of brain problems, Alzheimer's disease, for example, stroke patients, and other learning deficits. So what is neurogranin? I said it's actually a protein. It binds to this protein called calmodulin, and we think it mediates learning through environmental enrichment. And when it binds to calmodulin is sort of like a sink that stores calcium, and when it binds to this protein, it results in the release of calcium. And it's specific to the brain, which is very important from a drug development standpoint, because if we want to do something, ideally perhaps to boost up the level of neurogranin, you always have to worry about side effects in other organs, and so we don't think that would be an issue because it's not expressed anywhere except for the brain. And what's very interesting is if you go to the medical literature, there are over 200 peer-reviewed publications describing the function of neurogranin in animal models. And what's very clear is that we actually have the technology to specifically delete the neurogranin gene in mice, so analogous to your children. And clearly, mice that have either one or both copies of the neurogranin gene deleted not only have learning problems, but in talking to my colleagues who study this gene, these mice are very anxious. And you probably just heard a few moments ago Sarah's talk that it's very suggestive, and I think a lot of you would agree that anxiety seems to be very prevalent in a number of your children. So neurogranin, again, it's found in the neurons, and in particular in regions of the brain that we think are critical for memory formation and decision making, specifically the cortex and the hippocampus. And neurogranin is actually exactly in the part of the neuron called the synapse where the business is for mediating learning, and it's involved in memory formation. So this is kind of a cool picture that Andras provided of what a neuron looks like. This is, I think, from a mouse brain. And here's a little closer up view of it. So that central thing is the nucleus of the cell, and all these little outcroppings or whatever are the axons that have to make specific connections with other neurons from other cells. And it's an ongoing process, particularly in children, as they're learning, and actually through all of our lives, new connections are being made all the time. And this is a picture of what one of those connections is. That's called the synapse. So you have what's called the presynapse terminal, and then when that signal is transmitted to a connected neuron, there's actually a chemical mediator called a neurotransmitter that then binds to the other receptor there at the postsynaptic neuron or dendrite, and that's where neurogranin happens to be sitting that mediates this learning mechanism. Why is it not advancing? Okay. There we go. So, and if you look in the brain, for example, the hippocampus, there are bazillions of these neurons, and if you look up closer, they are organized in an amazing way that's critical for normal brain function. So, what is learning? It's something called long-term potentiation. So we learn through repetition, and as we learn more, what we find is that the neurogranin levels increase. And so you can have a minimal response, a short-term response, and with constant reminding, although my wife would disagree when it comes to reminding me to clean up after myself, but hopefully over time we develop a little better memory through repetition. So again, neurogranin is required to improve our ability to create memory and to improve long-term memory. So let me tell you now a little bit more about what we know about the function of neurogranin, at least in mice. So we can actually use our genetic engineering trickery to specifically get rid of either one or both copies of the neurogranin gene in mice. And remember, your children that have a deletion that spans neurogranin, you still have one copy of the they still have one copy of the neurogranin gene. Again, it's decreased there's decreased long-term potentiation in the postsynaptic dendrite, which is what I showed you where that neurotransmitter binds. And these mice have been shown to have a decreased ability to retain and recall memory. They have a decreased ability to perform cognitive skills, and as I mentioned, these mice are like all over the place. They're so anxious that it's actually hard to get their attention to learn. And interestingly, what my colleagues at the NIH that are studying this gene have shown is that if you take mice that are missing one or at least one copy of the neurogranin gene, they can actually respond to this so-called environmental enrichment protocol. And let me just kind of tell you what that is. So for a mouse, what makes an enriched environment? Well, if you put them in a larger cage with other animals and you give them lots of toys and actually you let them exercise. So these are all the things that a mouse enjoys to enrich their life. And when you do that, I kind of want to walk you through this slide, because this slide potentially is the basis for our approach to hopefully someday identifying a drug that might help compensate for the loss of one copy of the neurogranin gene. And so what I'm showing here, there's my arrow, is on the far left is knockout, which means those are mice that have both copies of the neurogranin gene eliminated. And the red is how they perform at sort of a baseline. And the numbers on the right side there are just sort of an arbitrary scale of their cognitive function. And if you just look at these first two bars, the knockout mice, so the red is the baseline, and if you take those knockout mice that have no neurogranin protein in their brain, if you expose them to environmental enrichment, they have no ability to improve. They don't respond at all. Now, very interestingly, if you look at the next two bars, the heterozygous mice, which means they have one copy of neurogranin gone and one copy retained. So that's analogous to the Jacobsen children. If you look at the red, they clearly have a higher baseline level of functioning, but very interestingly, if you expose those knockout mice to environmental enrichment, they improve in their cognitive ability, and that coincides with actually a boosting of that neurogranin protein to near normal levels. And even more remarkable maybe is the fact that if you take normal mice that have two copies of the neurogranin gene, here's the normal baseline level, that red, which is very similar to now the boosted levels of the heterozygote. And even mice, if you expose them to environmental enrichment, they boost up their cognitive function, coinciding with a boosted elevation or boosted level of the neurogranin protein in their brain. So that raises the very interesting question of, could we potentially elevate the level of neurogranin protein or somehow increase neurogranin function in the brains of, let's say, children that are missing one copy of neurogranin, but even in a broader sense, let's say patients with Alzheimer's disease. And it's been shown that patients with Alzheimer's disease, at least a subset of them, have decreased neurogranin levels in their brain. So how do we go about doing that? Here's the real question that we want to ask, is how far can we sort of push this out on the scale to see if there might be a beneficial effect? So the way we do that then is, well, here's our hypothesis. If we could somehow identify a drug or an agonist of neurogranin, could that improve learning and memory? And I already mentioned about Alzheimer's disease. And it could even have applications to other types of learning and memory problems, brain injuries, stroke, that kind of thing. So this is a neuron. These are neurons from the mouse brain, from the hippocampus, and all this orange is showing you what, the orange is showing you the neurogranin protein distributed throughout these neurons. And the neurogranin protein is actually coming and going to get to that part of the synapse that I told you about. And as an initial approach to doing this, we can actually use some genetic engineering trickery to basically introduce a neurogranin gene into the mouse. And this is just a cartoon that shows how we do that. We actually use a virus as sort of a vector to get the gene into the brain cell. And this is an experiment that Andras did last year. And I don't know how well you're going to see this, but basically all you need to appreciate is these two panels on the left are cells, brain cells from mice that have no neurogranin protein. So we've genetically engineered those mice to be lacking both copies. So the blue is just showing you the nucleus of those cells. And then below it, you don't see any orange because the orange is to show neurogranin protein and it's gone. And what Andras was able to do is, if you take the same cells and now basically infect them with that construct that I just showed you that has the artificial neurogranin gene, Now we see orange not only in the nucleus of the cells, but I know it doesn't project here very well, but you can see orange all the way out into those neurons, hopefully going to where neurogranin is supposed to function. All right, so I think I've told you about neurogranin signaling, how it results in actually electrical currents mediated by calcium. And it turns out we can actually take pictures of these electrical currents of calcium. And because of that, that gives us an opportunity then to develop an assay to screen for compounds that might increase this calcium signaling process. And this is probably not going to show up very well at all, but I'm going to give it a try. All this green that you see here is actually indicative of the calcium currents that are induced by neurogranin. And if I play, oops, that's not what I wanted to do. Now it's not coming up. But anyway, this was supposed to be a little bit of a movie clip, but actually when you run this in real time, what you see is little flickering green flashes. And those green flashes are actually the calcium signaling mediated by neurogranin. So if we take cells that have decreased neurogranin in a test tube, we could potentially find compounds that increase the amount of that flickering or that calcium current. And we can actually do that in a very high throughput way. So for example, I work with people at an institute here in San Diego called the Sanford Burnham Center, and a lot of drug companies are doing this as well. We can, in a relatively high throughput way, screen a library of 1 million different compounds where we don't know anything about the function, with the idea that we might be able to identify a compound that could be a potential drug that could be used to boost up levels of neurogranin protein in the brain. So finally, I told you before that we have these databases now available to us identifying patients with copy number variations, and in particular, there are several other genes that we've come across in the region deleted in 11q that look like they're also very important in causing some of the cognitive or behavioral problems. So the names of the genes so far are listed here: RIX, CURL3, and B3GAT1. I'm not going to go into their biology, but what's very interesting about these 3 genes is they all seem to have similar related functions in brain development. Specifically, they seem to have a critical role as the brain is developing in helping to make or mediate the exact synapses between neurons. All right, so just to summarize, the children that we've studied so far, you've heard this already in Sarah's talk, have variable degrees of cognitive impairment. Deletion of the brain-specific homeobox protein and neurogranin appear to contribute to cognitive impairment. Increasing neurogranin levels in the brain potentially could have very important therapeutic implications, and clearly there are multiple genes that contribute to the varying types of cognitive and behavioral problems that your children have. And I just wanted to finish by showing you this cartoon of the skin biopsies, why we're doing them. And up here on the top left, somatic cells, that means skin cells in this case. And with this genetic trickery, we can actually reprogram differentiated skin cells to become what we call a progenitor cell, or what we call a pluripotent stem cell. And that's sort of the precursor to a lot of different potential cell lines. And with that, then we can use further genetic trickery to reprogram those cells to become basically any cell that we want. So in this case, with my colleagues at the Sanford Burnham Institute, we can reprogram these stem cells to become neurons. And using your children, we can compare how those neurons appear in terms of their structure as well as their function to begin to pinpoint what the molecular mechanism is, not just in a mouse, but in your children. And that's ultimately going to be what we need to do to test whether these potential compounds or drugs can be effective before we can move to any kind of an animal model. And so obviously none of this is possible without a lot of people, particularly all of you in this room, speakers and patients and families. So Sarah and Natasha, goes without saying. Andras, who has really done a lot of the work that I showed you today. Dr. Huang at the NIH is probably the world's expert on neurogranin and has graciously shared with us a lot of the data that he has published and not even published about what neurogranin is doing. Many of you may remember, for those of you that were here 10, 12 years ago, our initial studies that began to establish a correlation between deletion size and cognitive function was by a psychologist here in San Diego, Dr. Zono Lai. Chris Cauldron and Teresa Mattina have been instrumental in helping us to do the deletion mapping, which is so critical because otherwise, if I had to pay for that, it would be about $2,000 a patient, which starts adding up when you're talking about 50 or 75 patients. Dr. Oga in Tokyo is actually someone who's been studying one of those other genes that I told you about and has graciously agreed to provide us with those mice that might allow us to get a better understanding of whether some of those other genes that I alluded to may also have a role in causing some of the cognitive and behavioral problems, particularly autism. So thanks for your attention, and I think we'll take a little break, Tim, while we try to set up. Do you have Matthias ready to go? We're good to go. Why don't we can we take a 5 or 10 minute break just to let people stretch their legs a little bit? But